everyone, this is Ragini Entry. Today we'll discuss on question and answers the topic Principles of Inheritance and Variation. So coming to the incurious things about this chapter, we have the chapter provides information about offsprings, how an offspring's genetic makeup is dependent on the parent's genetic makeup. That is, we have mother and father, which will give rise to an offspring. So the offspring's genetic makeup is dependent on the parent's genetic makeup. So this is what is explained in this chapter. So it also explains how Gregor Mendel analyzed the pea plant. So Gregor Mendel studied the pea plant to form the basics of genetics. That is why he is called the father of genetics. This chapter also contains the laws of inheritance, which was proposed by the Gregor Mendel, and it also explains about genetic disorders. The mark weightage for this chapter is like 8 marks, average of 8 marks for board examinations, and 4 to 5 questions asked every year. Topics to be covered is Mendel's laws of inheritance, inheritance of single gene, and inheritance of 2 genes. So which is like a monohybrid and this is dihybrid, and we have sex determination, mutation, and then genetic disorders. Coming to the first question, so we have to explain dominant and recessive traits. So explain dominant and recessive traits and tabulate the dominant and recessive characters in the pea plant chosen by Mendel. So first you have to explain what is dominant and recessive traits, and then provide a table of dominant and recessive characteristics in a pea plant, which was chosen by Mendel. So before going on to what is dominant and recessive trait, you have to know what a trait is. Traits are specific characteristic of an individual. So the individual can be an animal, human, or a plant. Here we take plant. So which Mendel chosen? So the dominant traits are those that get expressed in presence of another trait. So for example, we get tall and then dwarf. So if these both are together in the chromosome or in a plant, so this dominant gene gets expressed and it will suppress the recessive gene. So this is dominant trait, and recessive traits are those traits that are repressed by the dominant trait. So this small t is called as the recessive trait. So dominant trait are those are dominant, and recessive traits are those that are repressed. So this is dominant and recessive traits. So Mendel chose 7 characteristics. So Mendel chose 7 characteristics, and at first he didn't know what is dominant and recessive, and why after the experiments he did, he concluded what is dominant and what is recessive. And this is the chart showing seed shape, color, flower color, pod shape, pod color, flower position, and stem height of the pea plant, and its dominant and recessive traits. And these are the things that you have to be tabulated for the question. The next question is define true breeding. What do you mean by true breeding? True breeding is one that have undergone continuous self-pollination. So you have a tall plant. Let's say it is T, capital T, capital T. So at first you do not know whether it is capital T, or it is capital T, small t. Okay, so what you do is, because both of things will give rise to a tall plant, what you do is you self-pollinate. Continuous self-pollination will lead to a plant where only capital T you will be selecting. Okay, so So only the tall plants will be selecting and then self-pollinate it, and then at a stage you can confirm it as capital T because they won't give rise to dwarf plants. So that is called as true breeding. So and it is called true breeding line. So this is the definition. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Here, when T is self-pollinated, there is T cross T. So then they will give rise to only T, that is only small, that is tall plants. For how many generations you do this, only tall plants will be resulting. So that is what the same expression is obtained for several generations. That is to breeding. The next question is, when a tall pea plant is crossed with a dwarf pea plant, what is the outcome? Explain with Punnett square and ratios. So here we have tall pea plant. Here tall pea plant, T, which is the parent, and the dwarf pea plant, which is small t, which is another parent. As you saw earlier, these both have to be true breeding. So it is like T, for sure. It is tall. It is not like T, small t. It is T, this is small t. So when they are like cross-pollinated, we get gametes. That is, this is a plant with T, and they will provide us gametes. So gametes will segregate into T in one, another T in one, here small t in one, another small t in another gamete. Okay. So for first F1 generation, we are drawing the Punnett square. So make a square like this and then join these both. So T, small t, and then T, small t, and then here we will again get T and here small t, and then we are going like T and then small t. So here everything will be tall, but what is the difference? They are not pure, that is, they are heterozygous. They have T and they have small t. So but they are all tall. So to confirm, first they did a F2 generation, that is, they self-pollinated. That is T, small t selfing with T, small t. So we get gametes like this, small t, T, small t, and crossing this we get T, and small t and T, and then small t, and then capital T and small t. So here we see that there is So we see here that there is a T, one tall plant and two heterozygous tall plant. That is, we have three tall plants and then one dwarf plant. So this is like phenotype. Phenotype is something you could be able to see it. So this phenotype ratio is 3:1. So tall is 3 and dwarf is 1. And genotypic ratio is which is in the genes. So genes, we have one T, and then two T, small t. So we have one and two T, small t, and then we have this small t. So the ratio is 1:2:1. So you have to write first is the parents, and then the gametes, and then the Punnett square, and then the formation of F2 generation by selfing, and then Punnett square for F2 generation with the phenotypic and genotypic ratio of F2 generation. So this is the answer for the previous question. So the next one is based on the monohybrid cross. that we saw earlier. We saw here, right? This is monohybrid cross. Based on the monohybrid cross, Mendel proposed certain rules. So we have to state and define these laws. So what are these laws? The two laws are law of dominance and then law of segregation. So law of dominance says that in a dissimilar pair of factors, one member of the pair dominates the other. So what we saw in dominant trait and recessive trait, we have capital T and then small t. So capital T is dominating, small t is recessive. So this is what is stated in the law of dominance. So capital T dominates small t. This is law of dominance in a pair of factors. So that is here, factors means genes. A pair of factors, one dominates the other. So here capital T dominates a small t. This is law of dominance. And what is law of segregation? Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. So we saw in the previous slide, right? So we have this capital T, capital T. So we have this capital T segregating into gametes. So both the capital T will not come into one gamete. Only one capital T would come in one gamete, and another capital T will be segregated to another gamete. Same way here, small t will be segregated into two gametes. The same way it's here. So capital T, small t segregated into two gametes. So that is what? It is the law of segregation. So let's go to the next question. Here we have a question. Ria crossed the red flower, okay, with a white flower. So she expected a red colored flower because in the basis of dominating and recessive traits, Ria expected red flower to be dominating, but what she got was pink flower. So you have to explain why she got a pink flower. So let's get into the answer. The answer is because of incomplete dominance. So this is incomplete dominance. So it is a rule that does not follow Mendel's rules. Okay. So here we have parent generation, red flower cross white flower. You get capital R small r. Okay. This capital R small r, instead of being red, will be pink. Why? Because red is incompletely dominant. That is, when red and white both are expressed, but red suppresses white only with a small extent. So that is why you get all pink. And if you go for F2 generations, you get see this phenotypic ratio. We get 1 red, 2 pink, and then 1 white. So we get 1 red, 2 pink, and then 1 white. So this is the phenotypic ratio. So what we saw over here, Mendel's phenotypic ratio, it was 3:1. Okay. Here it is 1:2:1. The same as the genotypic ratio. We have 1:2:1. Understood? Okay, now next question. Here we have to explain the law related to dihybrid cross and the genotypic ratio thus obtained. So for monohybrid cross, we saw two laws. Laws of segregation, laws of dominance. And here we are going to see the third law, the law of independent assortment. So here, two pairs of traits are combined in a hybrid. Segregation of one pair of characters is independent of the other pair of characters. So here we have two pair of characters. So we have yellow round seed and then green wrinkled seed. So when the gametes come, we have a Y, capital R, and here small y, small r. So So here they state that the segregation of one pair of characters is independent of the other pair of characters. So this can be seen in the F2 generation. That is, we have capital Y, capital R, and we have small capital Y, small r, and they can be together. Okay, and they are segregated independently. So how they are segregated independently? You have capital Y, capital R, capital Y and small r also. So capital Y doesn't always come with capital R, and capital Y can also come with small r also. So that is, they are segregating independently. So if they are not independent, what happens? Only capital R will be formed all over, and capital Y and capital Y, capital R will be together, and small y, small r will be together always if they are not independent. Now if they are independent, so we get capital Y, small r and small y, capital R also. So they are independent of with respect to segregation. Next question is differentiate genes and chromosomes. Here we have genes. So genes is a segment of chromosome. So we have chromosome like this, right? So this is chromosome, and a segment of chromosome is the gene. Okay? Chromosome. What is chromosome? Chromosome is a condensed form of chromatin. So we have nucleus, and nucleus will have chromatins over here. So this is the cell, and we have the nucleus and the chromatin over here. This chromatin will condense to form chromosome. Okay? Genes. So genes segregate at gamete formation, and only one of each pair is transmitted to a gamete. So chromosome segregate at time of gamete formation, such that only one of each pair is transmitted to a gamete. So gene, we have two genes, right? Genes will have two pairs over here and over here. So we get like capital T and small t. So this is gene pairs. So they segregated gamete formation. So they segregated gamete formation as we saw earlier, and only one of each pair is transmitted to the gamete. That is, T goes to one gamete and this T goes to one gamete. And chromosome segregate time of gamete formation. They also segregate at the time of gamete formation, and one of each pair is transmitted to a gamete. So here we have like two chromosomes. So if we have two chromosomes, one of each pair is segregated to a gamete. Okay. Next difference is independent pairs segregate independently of each other. So this is similar to law of independent assortment. Here, one pair segregates independently of another pair. The same way, this if this comes, it doesn't mean even this also to come together. So this also like almost like independent assortment, but with the chromosomes. Okay. The next question is describe linkage and crossing over and how it varies from Mendel's dihybrid cross. So the answer is here we have linkage and crossing over. So linkage and crossing over. So crossing over is also called as recombination. So linkage, what is the definition of linkage? So two genes in a dihybrid cross are situated on the same chromosome. So we have chromosome. So two genes are situated in the same chromosome. This is like gene A, and this is like gene B. So they are situated on the same chromosome. The proportion of parental gene combinations is much higher than non-parental type. That is, like when parental is having like small a and then small b, there will be the offspring's recombination will always be small a and small b. Like majority of the offsprings. So this is linkage, that is they both are linked together, and it differs from the Mendel's law of independent assortment. In Mendel's independent assortment, what we saw, capital Y can come with anything small r, or capital Y can come with capital R. Okay, but when it is linked, we can say that small a will always come with small b for majority of the cases. So if this does not come with small a, or small a will not come with small b in a case called as recombination. So recombination is the exchange of genetic material, that is crossing over. The chromosomes, like two chromosomes, will cross over. So we have one chromosome here and we have another chromosome here, and they cross over. So in case if this has a capital A and this chromosome has small a, and when they cross over, like this segment comes here and this segment comes here, we have the tendency to get capital A, small b. Okay, this is like recombination. So these both rules will differ from the Mendel's law of independent assortment. So during Mendel's law of independent assortment, there is no crossing over or there is no linkage. But linkage and recombination do occur in other organisms. So they differ from Mendel's law, and you have to write actually linkage, definition of linkage, definition of crossing over, and you have to mention what is recombination and how they differ from Mendel's law. They differ from law of independent assortment. Next, you have to explain about sex-linked recessive disorders. So what are the sex-linked recessive disorders? They are color blindness and hemophilia. So here there is a characteristic. So for color blindness, there is mutation in X chromosome, and hemophilia, a single protein is affected and the blood will not clot. So blood clotting doesn't occur. Okay, this is color blindness and hemophilia. These both are sex-linked recessive disorders. So since they are sex-linked recessive disorders, they are transmitted from mother to son. Okay. So the next question is, explain the disorder that has trisomy of chromosome 21. So which disorder has trisomy of chromosome 21? It's Down syndrome. So Down syndrome. So this disorder was first discovered by Langdon Down. That's how the syndrome got its name. The affected individual. So how does the affected individual be? Short-statured, small round head, furrowed tongue, and partially open mouth. And the palm is broad and palm crease is present, and they have physical and mental retardation. So that's how the characteristics of Down syndrome. You have to mention what is trisomy 21 leads to Down syndrome, and then the disorders, how it affects the how the affected individual will look like. The last question of these is from pedigree analysis. You have to label these symbols. You get like square symbol is male, circle symbol is female, and if it's a diamond symbol, you do not know whether it's male or female. And if it's shaded one, the male female is affected individual. So, and if there is a single dash, it is a mating, and if it is double dash, it is mating between relatives. It is also called as consanguineous mating. And if there is a branch over here and with two more symbols over here, this called, they are parents. So these are parents and these are offsprings. Okay, So if the offspring is shaded, it is parents with the deceased child. So it has a round over here, it is a female child. If it has a square over here, it is a male child who is deceased. And the diamond shaped and the number in between inside we have, it indicates the number of offspring. The number indicates the number of offspring. So it is not shaded, so it is unaffected offspring. So the summary of this chapter is that the student will be able to answer Mendelian, non-Mendelian principles, and the student will be able to explain genetic disorders and terms such as multiple alleles, chromosomal theory of inheritance, linkage, heterogamy should be familiar with the student. This is about our institution, and for admission and other information contact this number.